affect the Hardy-Weinberg equilibrium, but we're going to work some problems relating to, well, first of all, just the Hardy-Weinberg equation, and then secondly, different types of natural selection. Are there any questions about what we discussed the other day about factors that can basically break Hardy-Weinberg equilibrium? One of the things we want to keep in mind when we talk about these factors, if you think about them, notice that mutations occur in real life all the time. So what this means in a sense is when we look at populations, they might be close to Hardy-Weinberg equilibrium, but they're hardly ever completely in Hardy-Weinberg equilibrium, just because of mutations. Even if nothing else is happening, mutations are a part of life. So from that point of view, they're going to happen no matter what. And at least so far, we haven't discovered any way to stop mutations from occurring. So we do want to keep this in mind when we look at the other factors, things like migration, natural selection. It's possible that there's no migration in or out of a population. So there's no gene flow. It's possible there's no natural selection, that everybody has the same chance to survive if we're looking at some particular characteristic. So we want to keep that in mind. If there are no questions, I'm going to go to screen sharing now, and you might want to have access to a calculator handy. So let's look at some problems where we just think about some of the basic math of the Hardy-Weinberg equation. And really the reason I gave you that one very short assignment about, heterozygous, homozygous, genotype, phenotype, things like that, we have to be able to think in terms of P and Q, our dominant allele and recessive allele, or whatever letters we want to use. But we have to be able to think, when we see a certain combination of letters or an individual letter, does that represent a dominant allele? Does that represent a recessive allele? Is it all the individuals in that population that show the dominant phenotype? Because when we work Hardy-Weinberg problems, we're going to be using those numerical relationships on a regular basis. So here we have a population of squirrels, and we have a dominant allele, big S, that causes dark colored fur. We have a recessive allele, little s, that causes light color. So if we look at this population, if we're actually seeing individuals in the population, and sometimes we'll just be given the statistics that resulted from someone else seeing the population. But if we look at this population, the first thing we have to figure out is how many squirrels are in this population. So if we count, we've got 3 rows, they've got 5 each, so that's 15. And then we have 1.
0083.
So we have 16.
Yes.
I think you need to mute whoever that is on the telephone.
Dollar amount, 1050 for 3.
Whoever's on the telephone, please mute. We can all hear you.
Alright, where's the next?
Okay, let's. So we have 16 squirrels in our population. 15 of them are dark colored. 1 is light colored. Now, remember what we said the other day. We're always going to start with individuals that show the recessive allele. So for the recessive allele, in this case, we have 1 individual that shows the recessive phenotype. 1 out of 16 squirrels is light colored. So that's going to be our starting point in this problem. And our starting point is always going to be, what frequency of individuals show the recessive phenotype. So the first question is, what frequency of squirrels show the dark phenotype? Well, notice for phenotype, this is just the dark color. So this is 15 out of 16. And you could leave your answer in this form, or you could change it to a decimal if you wanted. So you could put this into your calculator, and if you put 15/16 into your calculator, you get 0.9375. Now remember, one of the things we said about frequencies. Frequencies are numbers between 0 and 1. So when you figure out the frequency, not just in this question, but for any question, if you figure out the frequency and you get a negative number, or you get 1.3, that has to be incorrect. You have to have done something wrong, because the biggest the frequency can ever be is 1. The smallest it can ever be is 0. So 0.9375 is somewhere between 0 and 1. So that doesn't guarantee we have the right answer, but at least we know we didn't do something wrong in the sense that we got a number too big or too little. What's the frequency of squirrels that shows the light phenotype? Well, this is 1/16, or if we put it into our calculator, we'll get 0.0625. Now, notice we can do this simply by counting the fact that there's one light-colored squirrel out of 16 in the population. But there's a second way that we can do this. Remember what we said the other day. When we look at frequencies, the frequencies always have to add up to 1. So here there's only 2 possibilities. We have dark-colored squirrels, we have light-colored squirrels. So we could just take 1 and divide by 16, but there's another way we could do this. We could take 1, which is the total of all the frequencies for the phenotype, and we could subtract 15/16, the frequency of the dark colored squirrels, because they're either dark colored or light colored. So basically the light colored ones are what was left over. Or if you prefer decimals, you could take 1 and you could subtract the 0.9375. And you should get 0.0625. And notice you should get the same answer either way you calculate this. If the answers are different, if you're checking your work and you get different answers, then one of them has to be incorrect. Now, what's the frequency of little s? Show your work. Okay, so if we think about this, notice that what we really have is squirrels that are light colored are little s, little s. Their genotype is 2 little s's. Oops, that should be an s, not an a. So this means little s squared is 1/16. So if you remember, we said the other day, what can we do to figure out what little s is? Well, in math, if you have an equation like this, what do? You take the square root of both sides. So the square root of s squared, the square root of s squared is just s. And the square root of 1/16, if you don't remember how to take the square root of fractions, you can take the square root of the numerator, and then you can take the square root of the denominator separately. So the square root of 1 is just 1. The square root of 16 is 4. So 1/4, or You could also take the square root of 0.9375, or I'm sorry, 0.0625. And you would get the same thing. So you could have said, s squared equals 0.0625. And then you could take the square root of both sides. And if you do that, you'll find that s is 0.25. Now, one of the things to notice here, because it's something that's a common pattern that we see, notice there's only 1/16 of the squirrels in this population that show a recessive phenotype. But when we look at S, notice S is the frequency of the recessive allele. S is 0.25. A quarter of the alleles in this population are little s, even though less than 10% of the individuals show the recessive allele. How is this possible? Because most of the recessive alleles in this population are hidden. And they're hidden in some of these black squirrels that are heterozygous. So what's the frequency of big S? Now, big S, or the dominant allele, whatever it happens to be, we're always going to calculate the same way. We're going to find little s, the recessive allele, first, and we're going to subtract to find big S. So in this case, minus 1/4, and that gives us 3/4. And again, notice if we look at the two alleles, if we look at little s and big S have to add up to 1. That's because these are the only two alleles we're talking about in this population. Now, if there were 3 alleles, we'd have to know S1 and S2, and then we could figure out S3 by subtraction. But in this case, as soon as we know little s, we can subtract, we can find big S. Are there any questions about how we did our math here? Okay, let's look at another population. And sorry, these are homemade graphics, so I'm not much of an artist, so pretty much all our populations are black and white individuals because I can just fill in the picture and not fill it in. Okay, so we have a population of rabbits here. And big R causes the dark color, little r causes the light color. And notice in most mammals, the allele for dark coloration in the fur is dominant. So I didn't just pick that randomly. So again, first thing we have to think about is what is the size of our population? So we have 3 rows with 7 individuals each. So that's 21. And then I see 4 down here. So we have 25. So let's look at what our questions ask. What frequency of the rabbits show the dark phenotype? Okay, so we're just going to count dark individuals because that color we see, that is the phenotype. Okay, so I see 4 in the first row, 5 in the second row, that's 9. I see 4 more in the third row, that's 13. I see 3 in the last row, that's 16. So the frequency here is 16/25. Or if you like the frequency to be expressed as a decimal, if we divide, we're going to get 0.64 when we divide. Okay, now if we look at our population, there's only 2 colors. The rabbits are either dark colored or light colored. So we can count up the light colored rabbits, and there are 9 of them, which is equal to 0.36. But the other way we can calculate them, remember the problem we just worked a moment ago, is we can take 1 minus 16/25 and that's equal to 9/25. Okay, I'm gonna stop right here for a moment, and we're gonna take about 5 minutes, and I'm gonna let you work on questions 3 through 6 about these rabbits. So we figured out the individuals that are, if we think about the dark phenotype, that could either be big S squared, or that could be 2 times big S little s. Remember that Hardy-Weinberg equation. If we think about little s squared, that's equal to the light phenotype, or little r squared, sorry, in this problem. So you're going to have to start out, figure little r, then big R, and then you're going to have to use the Hardy-Weinberg equation to predict what frequency of rabbits are going to be heterozygous. And then, oops, I wrote down 24 up here. I should have said 25. And then how many rabbits do you expect to be heterozygous? So let's take about 5 or 6 minutes and work on this, and then we'll come back and we'll look at this as a group. So the clock is ticking. Use your calculator, see if you can figure these out. Think about kind of logically how you would analyze these questions. 3 and 4 should be pretty obvious. 5 should be obvious if you think about the Hardy-Weinberg equation. 6, you might have to think about a little bit. another minute and a half, and then we'll get together and we'll see if you have some solutions that you're happy with. Let me know if you need more time. And if you need more time, we can, I'll extend the time another minute or two, but otherwise we'll talk about the answers in just a second. Okay, let's see what you came up with. So let's look at the calculations that we would need to do. So notice number 3, what's the frequency of little r? Show your work. So in this case, we know that little r squared is equal to 0.36 because little r squared, remember, is the rabbits that show the light colored phenotype. So if little r squared is 0.36, we can take the square root of both sides of the equation and we'll get little r is equal to 0.6. Or if you're doing this with fractions, and at least for the first few problems we work, you will be able to do this with fractions. Remember what I said, little r squared is 9/25. So if we take the square root of the numerator, the square root of 9 is 3. We take the square root of the denominator, the square root of 25 is 5. So 0.6 or 3/5. And remember what we said, if you work this with fractions, you should get the same answer as if you work this with decimals, with frequencies. What's the frequency of big R? Okay, one of the things that you want to take home, kind of the take-home message here, to find the frequency of the dominant allele, pretty much we're always going to do it the same way. 1 minus the frequency of the recessive allele. So 1 minus 3/5, or if you prefer, 1 minus 0.6, I would accept either. So 2/5 or 0.4. Either one of those would be good. Oops, not comma 4 though. So one of those two would be the way that you would want to work this. And I would suggest that when we work our problems, the first couple, we're going to be able to use fractions. But later we're going to get to some numbers that are going to be a little more complicated. So you're probably going to want to use decimals to represent the frequencies. I won't take off if you don't, but it's just for some of those. You might have, something like 17 be your fractions for a numerator or denominator. It's going to get a little messy. What frequency of rabbits do you expect to be heterozygous? Well, remember the Hardy-Weinberg equation says that heterozygote frequency is 2 times big R times little r. So 2 times 0.4 times 0.6, and that's, whoops, not 48.48. Or again, for these first ones, if you're working them with fractions, it's 2 times 2/5 times 3/5, which is equal to 2 times 2 times 3, 12 divided by 25. Oops, 2 decimals there. How many rabbits do you expect to be heterozygous? Okay, now notice there's a difference. You might at first glance think questions 5 and questions 6 are the same, but think about why they're different. 5 is the frequency. 6 is how many actual rabbits are heterozygous. Now, one of the things to keep in mind when it asks you a question like number 6, the answer to that always has to be a whole number. You can't expect 12.3 rabbits to be heterozygous. Might be 12, might be 13, but it can't be 12.3. So when you see how many rabbits, you're always going to have a whole number as your answer. Well, here, what we need to do is we're going to take the frequency. So we're going to take 2 times big R times little r, but then we have to multiply by another factor. And that factor is the population size. So this is going to be 2 times 0.4 again, times 0.6 again. So notice this part is going to be exactly the same as we had up above for 5, but what's our population size? 25 rabbits. So this is going to be 2 times 0.4 times 0.6, or if you're doing it with fractions, this is 2 times 2/5 times 3/5. It still has to be multiplied by the population size. Notice in this case, the fractions are probably easier because notice we have 25 in the numerator. We have 5 times 5, 25 in the denominator. So our answer is just 2 times 2 times 3, or 12. Which makes sense because notice our answer up here, if we were working number 5 with fractions, our answer was that 12 out of every 25 rabbits would be heterozygous. Well, since our population size is 25, that means 12. But notice if our population size was 100 rabbits, then we would expect 48 of those rabbits to be heterozygous. So the frequency is going to be used to find the number of rabbits, but anytime it says how many rabbits in the population, or later we'll have some problems that say how many alleles are in the population, we're going to have to think about the size of the population. So if you're having trouble distinguishing them, think about a classroom. I could ask you what frequency of the students are females. And if I wanted to know the frequency that are females, the frequency of females would be equal to the number of females in the class divided by the number of females plus the number of males, the total number of students in the class. Suppose that 0.5, half of the students are females. Well, if I have a class with 10 students, there wouldn't be the same number of females as if I have a class in a big lecture hall at one of the four-year schools where I have 250 students. There might still be the same frequency. There might be 50% in either case, but the physical number would be different. Now notice we could also think about alleles. So suppose I asked you, how many dominant alleles are in this population? So to find the number of dominant alleles, let's go back to the frequency of our dominant allele. So the frequency of big R that we calculated up here was 2/5 or 0.4. Now we have to take our population size into account. So there's 25 rabbits in our population, but there's something else that we have to think about. If this isn't something that's X-linked or Y-linked, how many alleles does every rabbit in our population have? Can anyone tell me that? If I asked, if we were looking at a population of people, and I asked how many alleles are there for skin pigmentation, for whether you're an albino or you have normal skin color, how many alleles do you have for that?
So,
I'm sorry, what?
is it so?
2, 1 from each parent. So after we multiply by the population, we've got to multiply by 2 because everybody has 2 alleles. So whenever we're asked about the number of alleles, we have to use the frequency, but we have to think about the population size, and we have to multiply by 2. Now, in real life, we could be working with an X-linked characteristic or a Y-linked characteristic, and then we have to know the genders of the individuals. We won't do that, but in real life, you might have to. So we have 0.4 times 25 times 2, and that's going to come out to be 20 alleles. Now, think about this population overall. There are 50 alleles in this population, 25 times 2. Now, if there weren't any dominant alleles, there would be 0. But if everybody was homozygous dominant, there would be 50. So notice, if you think about the population size, if there were 100 rabbits in our population, the biggest answer you could get for the number of alleles would be 200. If you got something bigger than 200, somewhere you went wrong. You couldn't get something more than 200 because if everybody only has 2 alleles and there's 100 individuals in our population, there's only 200 total. So we have to keep that in mind. So are there any questions about how we figure out things like this? Now notice there is another way we could figure out the dominant alleles. It's much more work. We could take all the individuals who are heterozygous. So big R, little r, our Hardy-Weinberg term. And we could multiply that by the number of rabbits in our population. Because everybody who's heterozygous, every rabbit, has 1 allele. And we could add 2 times big R squared times our population size. Because this is everybody who's homozygous dominant. And all of these individuals have 2 alleles. So we could do this and we could add this up and hopefully we would come out with 20 alleles again. But I would recommend just take the allele frequency, multiply by the population, multiply by the fact that everybody has 2 alleles. Because the problem with this is, there's a lot more math. You're a lot more likely to make a mistake somewhere than up above. You know, that's pretty straightforward. The frequency of the allele, the size of the population, times 2, because everyone has 2 alleles. Now, in real life, biologists trap individuals like rabbits or squirrels. They look at whatever characteristic they're interested in. We have another problem with deer. We're going to skip beyond that and we're going to go to the next section to some problems where the counting has been done and we're just given numbers rather than having to do all the counting ourselves. So in humans, the little a allele is recessive to the big A allele. Little a causes albinism, big A produces normal skin pigmentation. In a small population, which is at Hardy-Weinberg equilibrium, 100 out of every 10,000 people is an albino. So what is the little a allele? What's the frequency? Okay, so we have 100 divided by 10,000. And if we have 100 out of 10,000 and we cancel 2 zeros, that's really like 1 out of every 100. So the frequency of the little a allele, you might think is this, but keep in mind what we're calculating here is little a squared, because this is people who are albinos. So being an albino is your phenotype. So albinos show the recessive phenotype. So what does that mean? That means that they have 2 recessive alleles. So little a squared is equal to 1 over 100. So if we take the square root of 1 over 100, what we're going to see is that little a is 1/10. Or if you're doing this with decimals, with frequencies, you'll be taking the square root of 0.01, and then little a will be 0.1. And again, notice something I pointed out before. This is a population of 10,000 people. 9,900 of them have normal skin pigmentation, but the frequency of the little a allele is 0.1. 1 in every 10 alleles is the allele for albinism. Why? Because most of these alleles are hidden in people who are recessive. Back in 1900, when Gregor Mendel's work on genetics was first rediscovered, a lot of biologists didn't really understand the idea that if you look at a recessive allele like little a here, little a is hidden in people who are recessive. So a lot of geneticists, after they first read Mendel and didn't really understand it thoroughly, leapt to a conclusion and did some really horrifying things. They started looking at people who were albinos or people who had different forms of mental handicaps, and they started sterilizing them. And what they thought was, let's think of albinism for an example. If we take all the people who are albinos and we sterilize them, in a generation or 2, we'll have eliminated this genetic disease from the human population. Well, besides the fact that there are obviously a lot of moral problems with this, notice that genetically, that's not going to work. Why doesn't it work? Well, if you look at most children today who are born as albinos, neither of their parents is an albino. Both of their parents have normal skin pigmentation, but both of their parents carry a recessive allele for albinism. And those two recessive alleles get together every once in a while. Remember when we did our Punnett squares in 105, if you have two parents that are heterozygous, we expect about 1/4 of their children to be homozygous recessive. So the problem is, besides the moral issues, if you sterilize all the people who are albinos today, that's not going to stop most albinos from being born in the next generation, because most children who are albinos are not born to two albino parents. So besides the moral problems, they didn't really understand genetics. Okay, now what's the frequency of the big A allele? Well, remember, we're always going to find big A. We're always going to find the dominant allele by taking one, oops, I wrote my answer down instead of writing down my, we're always going to take 1 minus the recessive allele. And again, if you were doing this with decimals, you would get 0.9. And again, notice as a double check when we're working a problem like this, little a and big A have to add up to 1, unless we have 3 alleles in the population. Then A1, A2, A3, whatever we're using as our symbols, have to add up to 1. What frequency of people in this generation are homozygous recessive? Well, the frequency of people who are homozygous recessive is our 0.1, the frequency of little a squared. Except, and we could work this out, if we multiply this out, 0.1 times 0.1 is 0.01, except notice 0.01 is 1/100th. That's really what we had up here. The thing that we took the square root of to find little a is little a squared. So in a sense, we could do the math, we could work this out, we'll still get the right answer. But the other thing we could do here is find the frequency that we took the square root of in A up above. How many people in the next generation will be heterozygous if the population size stays the same? Okay, so population size. So that means we're going to have to multiply by the population size. So this is going to be 2 times big A times little a. Times the population size, or 2 times 0.9 times 0.1. And it says the population size stays the same. So we've got to go up to our original information. So we're talking about 10,000 people. So if we do this, we have 2 times 0.9 times 0.1. And then we multiply by 10,000. And we're going to get 1,800, I believe, if I did my math correctly. So notice the questions in a problem like this are worked exactly the same as they were up above when we were looking at the pictures of the rabbits and the squirrels. The difference is, instead of us going through and counting all the rabbits and the squirrels, that's already done for us. So let's look at one more like this. In humans, the little p allele is recessive to the big P allele. Big P allows normal metabolism. Little p causes an illness called phenylketonuria. If you have any food with NutraSweet or aspartame in your house, if you look on the side of the label, it'll say something like, warning, phenylketonurics. This product contains phenylalanine. Because people who have phenylketonuria and eat phenylalanine, which is an amino acid that's found in proteins, they can have suffer brain damage. It doesn't usually affect adults. It mostly happens in babies when the brain is developing. So right after babies are born, they're tested to see if they have phenylketonuria. This is one of the first tests that newborns are given. So what's the frequency of the little p allele? Okay, so this disease is caused by little p, so that means it's recessive. So if we look at the people who have this, it's 256 out of every 10,000. So if we take the square root of this, we can leave it as a fraction, or we can convert it to a decimal. Oops. So keep in mind, this is the phenotype. So right now what we're talking about is p squared, little p squared. So little p squared equals 256 over 10,000, or 0.0256. We're going to take the square root of this. And if we take the square root of this, we're going to find that little p is equal to 0.16. Okay, well, big P is going to be 1 minus little p. Or 1 minus 0.16, so 0.84. And this is why I said, oops, forgot an equal sign there. And this is why I said eventually you want to start using decimals, because you know, the fractions are going to get kind of messy if you don't. I wouldn't take off if you stuck with fractions. Notice again, both of our frequencies are between 0 and 1. Whenever we're talking about frequencies, it doesn't matter what it's a frequency of. A frequency is always greater than or equal to 0, less than or equal to 1. No negative frequencies, no frequencies bigger than 1. Because 0 is 0% of whatever we're talking about. 1 is 100%. What frequency of people are homozygous for normal metabolism? Well, to be homozygous for normal metabolism, you have to have big P, whoops, and it has to be big P squared. So this is going to be 0.84 times 0.84. What frequency of people are heterozygous? So this is going to be 2 big P times 2 little p. How many people out of 10,000 are heterozygous? Well, it's going to be our frequency of heterozygotes, but our frequency times the population size. How many copies of the big P allele are there in 10,000 people? So it's going to be the frequency of big P times the population size. And then remember what we said earlier, everybody has 2 alleles. So we're going to multiply by 2. So 0.84 times what's our population? 10,000. Times 2. So notice the mathematics of this is the same as the ones we were doing at the beginning with the pictures. So are there any questions about how we've calculated any of the numbers that we've done? Okay, I'm going to stop sharing this screen. We're going to look at one more type of problem. And then I'm going to have you do a couple. Oh, that's weird. You guys see a green line down the middle of your picture? Oh, it's gone now. Can you see a picture of our peppered moss that we were talking about the other day?
Yes.
Okay, notice this is, remember what we said, they come in a variety of colors. And the peppered moss really is a good name for them. It kind of looks like somebody shook a salt shaker and a pepper shaker over them. Even the dark colored moss have some light speckles. Even the light colored moss have some dark speckles. But you can see here that depending on what background they're resting on, one of them could be much easier to see than the other. In this case, I would say the darker one shows up more clearly. So we're going to look at some problems with natural selection. Now, people who have studied peppered moss divide them into different color categories. I've looked at a couple of real colored peppered moss, and you must have to study them a lot more than I have. I've just glanced at a few, but they classify them into 9 different colors. So starting with number 1 is the lightest, it's almost white. Number 9 is the darkest, it's almost black. And then 2 through 8, the colors in between are various shades of gray. 2 is almost a very light gray, it's just got a few dark spots, almost a white. Number 8 is a very dark gray, almost black, it's just got a few white spots. So let's look at a chart, and our chart is going to have 6 rows on it. So actually 7, I guess, if we count the colors. So the 9 colors are listed up above. And then we have 3 different sections. We have totals, which is actually the number of moss, and frequencies before selection. We have totals and frequencies selected. And keep in mind, when we're talking about natural selection, individuals that are selected, these are moss that get eaten by birds, that the birds spot them and eat them. And then we have totals and frequencies after selection. Okay, so the first thing we're going to do is we're going to figure out what our totals are after selection. So we started out with 49 moths in our population that were the lightest color. 12 of them got eaten, so we're going to subtract. So we've got 37 left. We started out with 112 number 2s, 23 of them got eaten. So when we're done, we have 89 left. We started out with 147 in the number 3 category. We had 36 get eaten, so there's 111 left at the end. We started out with 211 number 4s, 57 got eaten. There's 154 left now. We started out with 262 in the category number 5. At the end, we had 72 get eaten, so there must have been 190 remaining. We had 209 number 6s, 59 got eaten, so there are 150 left. If you notice me make any subtraction mistakes, correct me. Number 7, we begin with 152, 41 get eaten. So we've got 111. Number 8, we start with 111, 28 get eaten. So we have 83 of this color left. Number 9, the darkest color, we have 42 at the beginning. We have 14 get eaten. So there are 28. Now, what we want to do when we look at this is ultimately we're going to have to try to figure if natural selection is occurring. And if natural selection is occurring, we're going to have to try to figure out what type of natural selection. So first thing we need to realize is we need these totals. We need to know the actual numbers of moths that were there before selection, that were selected, that were eaten by the birds, and how many survived, how many got away. But that's not what we're going to use to get our final decision. We're going to base our final decision on frequencies. So to find a frequency, here's what we have to do. Remember, we said that frequencies are basically fractions or percentages. So we're going to make these fractions. So I'm going to take my numbers down and I'm going to put the number in each box in the numerator. We'll find out what the denominator is in a minute. Be patient. Okay. So to find the frequency, if I were trying to find the frequency of males in our class, I would count the number of males and I would divide by the number of males and the number of females by that sum. Here, if I want to find the frequency of the number 1 color, I have to take the frequency of number 1, 49, and what I'm going to divide by is the frequency of 49 plus 112 plus 147 plus 211 plus 262 plus 209 plus 152 plus 111 plus 42. Why? Because this is the total number of moths in our population before selection. We've got to add all those up. Okay, I'm going to have to take a minute on my calculator now, unless somebody's a step ahead of me and has added all those up in the denominator.
1295.
1295? Okay, thank you. Okay, so we're going to divide by 1295 here, and we're going to divide all of these by 1295. So whatever number we divide one of them by, everything in this category is going to be divided by the same thing. And I think I'm going to have to put some of these numbers down on the bottom because my chart is expanding. Okay, now we're going to have to do the same thing for the number selected. So notice this was our oops. This is how we're going to do our before. So now let's think how we're going to do selected. And selected is really going to be parallel to this. So let's think of number 1 as an example again. Number 1 is going to be 12 divided by 12 plus 23 plus 36 plus 57 plus 72 plus 59 plus 41 plus 28 plus 14. 336 maybe? Let me know if you did the math and got something besides 336. Oh, oops. That should have been down in the frequency, not up in the totals column. Sorry about that. Okay, now notice we know the total number of moths in our overall population. 1,295. And we know how many of them got eaten, 336. So notice we don't have to add up our totals for the after selection. If you think about it, if we started out with 1,295 moths in our population and 336 of them got eaten, then really the total after selection is equal to our 1,295 minus our 336. Now, notice this is kind of based on the assumption that we've been doing all our math correctly. You know, if some of our math is wrong, we might have a mistake somewhere in here. So this is 959, it looks like. Keep in mind, if you've noticed any mistakes in the math as we go along, tell me. Won't hurt my feelings, I promise. Okay, now what we're going to have to do is we're going to have to compare these frequencies. Now, even though we start out with fractions, Clearly, if we look at these, we don't want to leave them as fractions. At least for me, I can't look at 49 over 1295 and compare that to 37 over 959. That doesn't really do much for me. So I'm going to get out my calculator and let's convert these frequencies to actual numbers. So let's look first at number 1 before frequency. And before frequency, we have 49 divided by 1295. And I'm going to round this off to the 3rd decimal place. And the reason we want to round off to 3 decimal places, you could do 4 if you wanted. But a lot of times with natural selection can be important if one particular phenotype has a 1/100th advantage. So you don't want to round off too carefully. So let's look at 12 divided by 336. Let's look at the number ones that were selected. And that's point. And I got 0.036 when I did that. Okay, and then for the last one, we're going to look at 37 over 959. Wait a second, how did I? Okay. Let's take 37 divided by 959. And I get 0.039. Now, notice that these 3 numbers are not the same. What we have to do is we're going to have to compare. Before with selected or before with after. You can do either one. Now, what we have to keep in mind is what I've said about what these 3 categories represent. So before is all the moths in our population before a flock of birds flies through the forest where they're hiding on trees. Selected is which unfortunate individuals, the moths, get spotted by the birds and get eaten. And then after selection is the flock of birds moves on, who remains? So let's think about, first of all, comparing before with selected. Let's take a more dramatic example. These numbers are all pretty close, but let's think about the general case. Notice that in this case, selected is a smaller number than before. If selected is smaller, does that mean that the number 1 colored moths are more likely to get eaten by birds or less likely? Notice that if we look at the population, 38 out of every 1,000 moths, rounded to the third decimal anyway, 38 out of every 1,000 moths are color number 1. If we look at who gets eaten, 36 out of every 1,000 moths are color number 1. So notice color number 1 must be slightly hidden, because really what we would expect is if color number 1, suppose there's no advantage and the birds can see each color of moth equally. Well, then if there are 38 number 1s in every 1,000 in the population, there ought to be 38 number 1s that get eaten. But there aren't 38, there's only 36. So when we're comparing before with selected, if selected is lower, then that means that that category, whatever we're talking about, in this case, the color, is more likely to survive. If the selected is higher, then that category is less likely to survive. Imagine that our number 1 moths only make up 38 out of every 1,000 moths in the population, but suppose that 900 out of every 1,000 moths that got eaten were colored number 1. That would mean that number 1 really showed up. The birds were able to find it, or maybe they're not good flyers. Maybe the color isn't the whole story. So if this number is lower, that means that number 1 is more likely to survive. Now, notice that if we're comparing before with after, and I think this is a little more logical, but you know, that depends on you as an individual. If we're comparing before with after, if the after is higher than before, and when I say after and before, I'm talking about the frequencies. If the after is higher, that category is more likely to survive. If the after is lower, if the frequency goes down, if it starts out and 90% of the moths are color number 1, and after selection, only 20% are number 1. So if the frequency after selection is lower, then that category is less likely to survive. So here's what I would like you to do for now, because we're going to look at these problems tomorrow. So I would like you to finish calculating the frequencies for the other 8 colors. And you're going to calculate the before and the selected and the after. And for each color, so 1 through 9, because I don't think I told you for one. I tried to avoid saying it, but maybe I did. For each color, 1 through 9, what I want you to do is tell whether that color is more likely to survive or less. And notice it could be that their odds of surviving are equal. It might be that 0.2% of the moths are number 4 before selection, and 0.2 are selected, and the frequency after selection is 0.2. So nothing changes. So if nothing changes, that basically means that's kind of what we would expect. We would expect that if this is just kind of if you think of it like a grab bag and the birds are just picking moths randomly, if color doesn't affect them, then if 20% of the moths are one color, the birds ought to eat 20%. If 19% are another color, the birds ought to eat 19%. So if the birds eat more than we have at the beginning, that color is less likely to survive. If they eat less, they're less likely to survive. Now, there are a couple of questions that go along with number 1, but for now, I just want you to fill in the rest of the chart. And down at the bottom where it says, you know, the frequency after selection, tell me in that cell whether they're more likely to survive or less likely to survive. So don't worry, there's 2 questions down below, number 1 and number 2. For now, you don't have to do those. We'll talk about those, well, tomorrow. So any questions about how you're going to fill in the rest of the chart? Now, I'd like you to do 2 other problems as well. So on the first worksheet, well, and actually, I think just one other problem. I want you to work the deer and that's the next diagram on the Hardy-Weinberg problem. And see if you can go through the answers and figure those out. And for now, I'm not going to collect either of these, but I would like you to work on them by tomorrow because we'll talk about the answers. And then we'll have some for next week, probably Monday, but maybe Wednesday that you will have to turn in. So any questions about what you're going to do? So you're going to finish up this first problem here, number A or letter A that we've started on. And you're going to go back to the other questions and work the next diagram, which is a picture of some deer. And basically for the other frequencies here, you're going to use the same method that we used for number 1. And keep in mind when we talk about, you know, comparing before and after, you're always going to be comparing number 1 before and after, or number 1 before and selected. Don't compare number 1 before with number 2. We'll talk about why we might want to do that tomorrow. But for now, I just want you to calculate the frequencies for the rest of the chart, round off to the 3rd decimal place. So don't just truncate your answer, round off. And then at the bottom in that last cell, write down if that particular color is more likely to survive, less likely to survive, or if you find any that are just the same frequencies all the way through, you can say same or equal. Any questions? Okay, if not, I'm going to let you go, and I will see you tomorrow morning bright and early, and enjoy the rest of the day.
Thank you. Have a good day.
You
You too. Bye-bye.
Bye-bye.
Have a good day.
Take care.
You too.